I think it's a really important time in osteoarthritis drug development. I think that we have really seen a movement in the context of the science to understand what's going on. And much of that work has been done both academically as well as within the industry. Furthermore, we've seen a real move by the regulatory bodies around the world to consider unique ways of approaching this incredibly complex disease. And although the guidance documents are still somewhat behind the times, I do think that there has been significant progress. Beginning in 2008, the world responded to an FDA RFP about how to do better in studying drugs, both from a palliative point of view, meaning signs and symptoms improvement, as well as perhaps thinking about structure modification in osteoarthritis, altering the natural history of the disease. That's obviously the holy grail. We'd like to be able to identify that patient population or that individual patient who will likely develop a disease that's not only painful, but also mechanically abnormal, leading to destruction or alteration of function of the entire joint. This then led to OARSI getting very involved in trying to figure out how to maneuver a complicated regulatory environment, since basically drugs are approved both in Europe and in the US and elsewhere about how a patient feels, functions, and survives. That critical bit of evidence suggests that if a drug is to alter the natural history of the disease, altering the structure of the total joint, it also has to have symptomatic benefit associated with what is a measurable change. This has been complicated, and also complicated by who are you going to study? Are you going to study people with late-stage disease and accrete new cartilage and thus improve the structure? Or is that not enough? And do you have to address also the subchondral bone? And perhaps that is not enough. And then the other question is, do you want to prevent the progression of disease, or do you want to progress the progression of onset of osteoarthritis? It's important to remember from a regulatory point of view, prevention means the patient doesn't have disease yet, or the patient is not progressing. And how to identify that patient population who is progressive or who will rapidly progress in the window of opportunity of measurement has always been a significant challenge. So we actually have a wonderful panel of people who have been working in the field for a while, some of whom are looking at the whole joint, some of whom are actually exploring a cartilage change, some of whom are looking at even earlier time sequences, some of whom are looking at the effect of senescence on the destruction of the joint. So Nancy, why don't we go back to our presenters and have them talk to us?
Very good, very good. Thank you so much, Lee, for that perspective. As I mentioned earlier, you have spent quite a bit of time in this space, both trying to help individual efforts related to compounds and helping the FDA to try to understand how best to assess compounds that might change the course of this disease. So I know as a organization or those with osteoarthritis, everyone thanks you. And on that note, let's start with having Dr. Tambiah talk to us about the work they're doing on at Samumed related to Wnt modulation and osteoarthritis. Thank you.
Hi, hopefully you can see and hear me okay?
Yes,
Great.
both.
Great. All right, well, thanks. And thanks for inviting me on behalf of Samumed. And that was a great introduction from both Lee and Nancy. So at Samumed, I head the medical affairs department. And so we are involved with all the departments within the company in helping the studies progress. Our compound is a CLK-DYRK1A inhibitor, and it modulates the Wnt pathway. And I'll show you, let's see if I can show you a slide here. Just let me know if you can see this.
Yes, we can see it.
Okay, great. So this really is a cartoon of the mechanism of action, and so the drug actually works as on a pathway which we know is upregulated in osteoarthritis progression, and that's the Wnt pathway. And the drug is a small molecule, and it penetrates the nucleus of adult stem cells, which are going to either in osteoarthritis form bone as opposed to cartilage, and by inhibiting these intranuclear kinases, changes the gene expression of them such that the sulfate of those cells become cartilage cells, or get driven towards cartilage instead of bone. Now, this is where we've been focusing our study. We know that the Wnt signaling pathway is involved in all the tissues of the joint, and so we believe that there are other effects going on as well in the other tissues. We just haven't, and we're learning more and more through our scientists. We just haven't got sufficient data together to be able to share that externally. So we're continuing with that work within the company. Let's see if I could just try to advance this slide here. Here we go. So there's a number of effects that happen. I'm not going to go through these in great detail, but basically, as a result of inhibiting these kinases, and because we know it affects more than one tissue, we also know that it has anti-inflammatory effects. It has effects on cartilage in terms of we think it can form cartilage chondrocyte regeneration. It certainly has done in vitro. We're yet to see that, to prove that in vivo. Cartilage protection, anti-inflammatory properties, and also when the drug is in the joint, it stays in the joint from its formulation, and it lasts in the joint up to 180 days and does not leak outside the joint into the plasma. And what we see also, if you look on the bottom of these mini slides, that there are certainly in animals, we see that there's very beneficial effects on the joint. And the red staining on the left is this osteoarthritic mouse model, and there's degeneration of the cartilage. And if you look on the right-hand side of treated joint, you can see regeneration of the cartilage and a lot of the joint tissues as well. So they're all these sort of preclinical animal work. Of course, what counts is what we see in man. So we did a phase 2A study initially, and we identified that subjects with unilateral symptoms showed improvements in pain and function. And then we tested that population in a phase 2B study as the ITT population, or full analysis set. And here you can see that out to, this is a single dose, this is the dose that we're taking into pivotal studies, the 0.07 milligram dose. And that we can see that out to 24 weeks, there was a signal, a pain signal, and bone function signal compared to placebo. And this was significant at 12 weeks, which is our proposed primary endpoint in the phase 3 studies. So in terms of structure, that has actually proved to be very difficult. Sorry, I'm just stopping sharing now. And that is because it's very difficult from the manufacturer's perspective. I'm sure we're going to hear from my esteemed compatriots out there from the corporate world to prove that a structural change in osteoarthritis is linked to benefit to the patients. And we've had some early radiographic data that is a signal that our drug slows down narrowing of joint space with. but we will have to do further studies to prove that more definitively. And also, we would have to link that with the change in symptoms that we see. And where it's unclear from a regulatory path what that entails. And I'm sure we'll get into more discussion of that as time goes on. But in summary, that's really a snapshot of our molecule. So it's a single injection into the joint, small molecule, WIT inhibitor, and it has effects on pain through anti-inflammatory properties. And from our preclinical studies, we believe it is structure altering as well. And currently we're in phase 3 studies, and we are intending to submit NDA at the end of 2021 if all goes to plan with the recruitment for our studies. So I think that I'll stop there. I think that's a pretty brief overview of our program.
Okay, well, thank you very much, Dr. Tambiah. And I think what we'll do is Dr. Simon and I will go ahead and let everyone give a short talk as you did, an overview, and then have a panel, everyone join the panel for discussion at the end. Is that, Lee, you good with that? Yeah? Okay, so Dr. Deckx, will you please share with us what your group has been doing in osteoarthritis? Thank you very much.
Sorry, Nancy, what was it to me that you were asking to?
Yes, Deckx. Yes, Deckx. Yeah, Henri.
yeah.
Henri.
Yeah.
Yes, please. Please. Sorry.
Yeah,
sorry.
it's yeah. Okay.
Welcome from. By the way, welcome from overseas, and good evening. Right? Sorry. Okay.
Yeah. Good evening and good morning to some of you. Hang in, I have to start my video, but I'm also going to share. I'm going to share some slides as well. After having thanked the Arthritis Foundation for inviting me to this panel, and thank you for your introduction, Nancy. And I'm very glad to be there and to share some of our data. So yes, I am a clinical development leader in a company called Galapagos. It's a young company, fast growing, originally specialized in target identification, but some of the targets we continue to work on and identified molecules that were active on these targets. And so in co-development with a French company called Servier, we developed our drug GLPG1972. Servier called S2010-86, but I'm going to talk about 972. It's the same drug. A molecule that in a potent way and selectively blocks ADAMTS5. And ADAMTS5, do I share my slide? Not yet, I think. Hang in there. I'm going to share my screen. There you go. Just tell me if you can see my slides.
Yes, perfectly. Thank you.
Okay.
Thank you.
So yes, so 1972 is a molecule that selectively and quite effectively inhibits the activity of ADAMTS5. And ADAMTS5 is an enzyme that cleaves an essential component of the cartilage called aggrecan. So it's an aggrecanase, and it cleaves it at a very specific location, producing neoepitopes that are identifiable in synovial fluid, but also in serum from patients. This neoepitope is called ARGS, and it is increased in patients with acute osteoarthritis or patients after or after traumatic event in joint or after knee replacement. And so we developed this drug, and we showed an anti-catabolic activity of this drug in vitro in cartilage explants from mouse and human, and with a clear dose response. This anti-catabolic was nicely shown, and then in the next step we showed in vivo in 2 validated preclinical models, the rat meniscectomized rat model and destabilized meniscus model in mice. We showed again with increasing doses and increasing disease-modifying activity on this cartilage. And then, to make a long story short, we started phase 1, first in human, other phase 1 studies, and then ultimately phase 1b study in osteoarthritis patients. It was a small study. It was our first study IND opener in the US, and I'm here in the right lower corner of my slide, where we, among other parameters, followed these famous ARCS levels in serum of the patients while they were treated with an oral daily dose of our drug with different dose levels, going from 100 to 300 milligram and compared to placebo. And as you can see on this graph, the ARCS levels in the serum of these patients nicely went down. It's not really a clear dose response, but a dose proportionate, meaning for the smallest dose we went down up to minus 40%, and for the highest dose around 55%. And this remained as long as the patients were dosed, and then as soon as they were discontinued from the drug after 1 month. It was a short and small study. It seems that the ARCS levels get back to baseline values, and you see also the orange line on top of the graph is the placebo, and so you see that there's a very significant difference between the active treated patients and the placebo group in terms of ARCS levels. So the conclusion of this study was that with our drug, we clearly were on target. We inhibited ADMTS5. We never made a conclusion that we inhibited cartilage degradation, which is the consequence of cartilage loss, which would be the consequence of losing aggrecan in the extracellular matrix of cartilage. Because ARCS is, let's say, aggrecan is in multiple tissues in the human body. ADMTS5 is present in multiple tissues, and what we measure in the serum is the activity of ADMTS5 on aggrecan in the body, not specifically in the joint. Nevertheless, based on these encouraging results, also based on an overall favorable safety profile of our drug, we decided together with Xavier to set up a large phase 2 study. Going to the next slide. A large phase 2 study that had a very classical design with a screening period, 5 weeks, 52 weeks treatment period, where patients were equally randomized to 4 dosing groups, 3 dose levels of active drug, 75, 150, and 300 milligram, and 1 placebo group, and then a safety follow-up period at the end of the trial. And when we designed this trial 3 years ago, we were pretty confident that by showing a difference in cartilage loss based on quantitative MRI of a specific zone of a specific anatomical location in the knee of these patients, that we would be able to demonstrate an effect of our drug, and probably as well, hopefully show some clinical benefit for these patients. And therefore, we tried also to enrich our population by selecting patients based on a fixed flexion radiograph of their knees, and selected the target knee based on a combination of two radiographic severity criteria, one based on the Kellgren-Lawrence grading, and the other one based on the joint space narrowing atlas from Altman pictures. And so we selected specifically patients with an KL grade 2 or 3 and joint space narrowing severity grading 1 or 2, in the hope to have patients that are in a certain severity degree based on data that shows that if you have a certain progression in a disease, you are more likely to progress in the following years as well. So that was the way we tried to enrich our population, and therefore we had QMRI at baseline at week 28 and week 52. So this study was started sometime in 2018. We had faster than expected inclusion, despite important screen failure rates based on this selection criteria. But we were quite successful in selecting the right patients, because in the end, we had a population with, let's say, 70% of the patients having GSN severity of GSN2, and almost 90% of the patients had Kellgren-Lawrence grade 3 knee being studied in this study.
Henri,
So,
Henri,
yes.
this is Nancy. Great. Could you put your slides onto slide mode?
Nancy, I'm losing you.
So she's talking about full screen. On the bottom, if you click
we see the full
The
picture.
slideshow mode?
We've got your slide, but we've got, and on the left-hand side, we've got all your slides, dear.
Yeah.
Yeah, full screen. On the very
Full
bottom,
screen.
right. I can't do it. Bottom.
on the bottom of
Oh well.
on the bottom of your
go
slide,
ahead.
you click the monitor menu. You take the monitor icon. Yes.
You want the full slide? That's what. That was the question.
Yes, sir.
Okay,
yes,
sorry.
Yes.
Okay.
No, perfect. Thank you so much. Okay, onward.
But yeah, so I don't know how much time I have. I tried to give you, let's say, an overview of the drug, how it works, and where we are recently. So this large phase 2 trial has ended. We had a last patient in the summer of this year. We had our top line results in October this year, and we published a press release with the conclusion that we unfortunately did not reach statistical significance on our primary endpoint. Our primary endpoint was the difference between these active groups and placebo in terms of cartilage loss in the central medial compartment of the between femoral and tibia. And this, that was the conclusion of our study. In addition, we had not only these primary endpoints, structural endpoint, we had also a number of secondary structural endpoints, as well as clinical endpoints, the classical WOMAC scale, the pain, VAS, the PGA. We also had calculated the ORC responder rates. All these results are very consistently showing that there is no or insufficient effect to reach statistical significance. So, and this was also mentioned in our press release recently. I think that's the main message I can carry so far. I think I can stop with my presentation here.
Perfect, perfect. That was wonderful. That was just wonderful. Great. Thank you for showing us all the data. Okay, stay with us. And now we're going to switch to Dr. Ben Zhu, who will tell us about a senolytic development program that was undertaken by Unity Pharmacy in San Francisco. Welcome, Ben.
Hello.
It does help us if you can put your slides onto full screen, if that's possible. We should do it at the beginning. So,
Yep.
And perfect. Thank you, Ben. Thank you very much. And Henri, I apologize for having to stop you in the middle of your talk, but this really helps everybody to see the slides a lot better. Thank you.
Sure, no problem.
Hi, Ben. You're in presentation mode. That's what we're seeing.
Yeah. Which one is it? It's not that. Here it is.
I think if you right-click on that and say hide presenter view, that might work. If you right-click on your screen.
Oh, just somewhere. Okay.
Yep.
That works.
That's it.
Okay.
Yep.
Great. Okay. So I'm going to tell you a little bit about UBX0101. This is a senolytic that was in development at Unity Biotechnology until recently, and the goal was to develop it as a treatment of knee osteoarthritis. Since I'm no longer at the company, as very recently, I'm going to just present to you kind of a generic slide presentation, not with any kind of company confidential information. But so UBX0101 is a MDM2 P53 interaction inhibitor, and what it was found to do in preclinical studies was that it was able to induce the apoptosis of senescent cells. The company Unity Biotechnology is seeking to develop therapies to address diseases of aging, and leveraging the biology of aging, first and foremost, going after this cellular senescence process. So when cells undergo stresses, or after they've accomplished a certain number of proliferative cycles, they can undergo a cell cycle arrest, initiating the cellular senescence process. And when they do so, they arrest, and then the cell sort of stays around and tends to express these proteins such as P16 or P21 to maintain the arrest. And they also secrete these senescence-associated secretory phenotype. It's a bit of a mouthful, and some people shorten it to just calling it SASP. But the SASP can include, depending on the cell type or the tissue that you're looking at, the SASP can include inflammatory factors, profibrotic factors, and growth factors. So the cells are, with aging, these cells appear to accumulate in tissues and just are putting out these factors locally. In the osteoarthritis field, we are kind of familiar factors that could be responsible for the structural degradation of the joint. So with this premise and with this mechanism, UBX0101 was further developed in some preclinical studies that seem to show good effects in the ACL tear injury model in rodents, and improved both the ability to weight bear on the knees, as well as decreasing the amount of degradation going on in the knee at the histological level. It also was seen to remove P16 positive cells, so this marker of senescence. You could remove them from synovial explants, and it could also remove cells from chondrocyte cultures in 3D. So armed with this kind of preclinical evidence, the company proceeded into filing an IND and to do a phase 1 study with UBX0101. This is going to be, the idea would be that this could be a single injection, could eliminate the the senescent cells in the osteoarthritic knee, and thereby kind of restore, at least reduce these cells which is creating these fast factors, and allow perhaps restoration of structure, at least to halt the amount of structural degradation going on. So a phase 1 study, a typical phase 1 study was initiated, typical single ascending dose study where you start at a low dose, injection into the knee, and then you, by successive cohorts, go up to higher, what you expect to be a high or at least a therapeutic effect, to check for safety and tolerability in the study. So that study was completed last year, June 19, 2019. We had very interesting data that we presented then as a late breaker abstract at ACR of the phase 1 study. Here's a table from that abstract showing that there were surprisingly effects over this 12-week phase 1 study, effects on pain, both by the WOMAC-A, the numeric rating scale, 11-point numeric rating scale, and the WOMAC-C function. So the placebo groups, you can see, did have some level of improvement, if you will, on these measures, a placebo response, but not a large one. And then there were, with the lower doses of UBX0101 in the center column, there were no perceptible real differences from placebo. However, with the higher doses that were tested in the higher dose cohorts, 1, 2, and 4 milligrams of UBX0101, this is 18 patients pooled, that there were some sometimes rather large effects suggested by this that were even statistically significant in this study, which was not powered for to show efficacy, but mainly a safety tolerability phase 1 study. So these were surprising and very encouraging to us. As I said, the main thought was that this would be more for a structural stabilization or improvement over the long run, rather than symptomatic improvement. But this gave us some encouragement to try to assay, evaluate the symptomatic improvement as well in further studies. So then a phase 2 study as I said, this phase 1 study was not powered to assess these kinds of symptomatic endpoints. But encouraged by this, we set out and did a adequately powered study, phase 2 study of 45 patients per arm with 3 different doses of UBX0101 versus placebo, a randomized control parallel group study, very standard kind of design, and conducted that pretty rapidly in the last year. Despite the pandemic, and recruited patients in the US, 183 randomized patients. And then what's the answer? So then the final outcome, however, was that the study was negative, that the 12-week, we did not meet the primary endpoint, which we set out to be the WOMAC-A, the WOMAC pain subscale improvement compared to placebo in any of the UBX0101 dose levels. Did not show an effect. And furthermore, when you looked at the pain by numeric rating scale, the NRS, or by WOMAC function, there were no differences. Another thing that was interesting to note out of this study was that there was a really large placebo effect in this, such that when we initially got these 12-week results, it was quite difficult to discern whether or not there was any kind of treatment effect whatsoever for UBX0101. It was just such a large one. And when we actually had done some research with a collaborating vendor, and determined that our placebo response in this study was actually on the high end, on the top 90% scale for placebo responses in osteoarthritis trials, including osteoarthritis trials with intra-articular drugs. So, but since that time, we had done further analysis of the 24-week data and of another study that was run, another phase 1b study, and really came to the conclusion that the drug at this regimen, single dose up to 4 milligrams, did not have a perceptible treatment effect, a detectable treatment effect. And therefore, the sad conclusion that the study program was going to be terminated, because from a business standpoint, this is just going to take too much to try to slog it out and further develop it. And the company has changed its direction strategically to therapeutic areas of ophthalmology and neurology, and away from musculoskeletal diseases. So that's the story of UBX01 in the last 2 years.
Well, thank you, Ben, for catching us up on that. And it'll be interesting in the panel discussion, to see other comments. Thank you very much. And now I'd like to hear from Dr. Christoph Ladel, who's been working, who did a lot of work or organized a project with recombinant FGF18 Splifamine for the treatment of osteoarthritis. And with Christoph, if you could tell us a little bit about that, would be great. Thank you.
Well, thanks for the invitation here to this session. Before starting the story about Splifamine, I was with Merck and I was project lead, so I can tell a little bit about the preclinical and also clinical story of Splifamine. But what I think is upfront, what I want to make the point, what we hear here in this session is we are talking to each other, and we are not talking to each other as competitors from the different companies. Everybody is struggling with the same problems, and we have this really great opportunity in the community, in the osteoarthritis community, to have this exchange of informations and lessons learned. And I think this will help in the future to bring something forward. One common theme in what I see, and we can discuss this in the panel discussion later on. One common theme is if we go for the all-comer populations, and most of the studies performed by Merck, Unity, Samumed, started with an all-comer population, and very quickly the companies learned that we are not addressing the all-comer population, but different segments. So for the organizers, they have done a great job to select the different areas. If it's anti-catabolic, like Henri has alluded to, if it's anti-senescence, if it's an Wnt inhibitor, or an anabolic factor, like I'm talking very briefly about this. I try to share my screen and hopefully it works. And go also to the presentation
Yes, perfect.
mode.
You got it. Very good.
And presentation mode is also working.
Chris, I think you also need to just right click and hide the presentation view.
Hide presenter view. Is it better now?
You're back into the you're not on full screen.
I'm not on full screen. That's a bad thing. How can I do this? I need to stop and go to full screen, most probably directly here on this, and then sorry for this. Does it work now?
Yep, looks great.
Okay, I'm talking and I share with Ben the point I have left Merck in this. Since Merck has taken as Unity a strategic decision to step out of osteoarthritis as a therapeutic area. There was a rather big group working on osteoarthritis with different projects, but Merck has, for strategic reasons, decided to step out. So I also stepped out, since I'm very much liking to be in the field. I've spent the last 17, 18 years in this field. Like I said, I was project lead of Splifamine there. Therefore, I decided also to leave the company. But the story will go on. That's something I can promise in this. Talking about Splifamine, switching completely gears here, since we are talking about an anabolic agent. What is the mode of action of Splifamine? Splifamine, in the first instance, triggers chondrocyte proliferation, and subsequently, these newly produced chondrocytes start to produce healthy matrix molecules. You have seen from my title, I'm PhD, so I'm coming from the preclinical part. And we have seen this in different settings, in the preclinical settings, that we are regenerating the cartilage, we get more chondrocytes, we get more extracellular matrix. And you see some in the bottom of this slide, you see some of the data. These are porcine chondrocytes, but we see the same with the human chondrocytes. We see a dose-dependent increase of proliferation of these chondrocytes. And the one thing what we have observed in the setting, the chondrocytes are not losing their phenotype. If you simply look through the microscope, they are not becoming fibroblast-like chondrocytes, but they keep their rounded shape, and that's in a dose-dependent way. So that was really interesting, and I will keep the story short. We have looked in different models, animal models, in rat, mouse, guinea pig, dog, horse, what have you. I think we are missing only a few of the animals. And we have looked also in the setting. Next slide. Where's the next slide? Next slide. We have looked also in the species human in this. As of today, roughly a little bit more than 800 patients have been exposed to patients. And I've seen on the call on the participants a lot of collaborators that have worked on this. So a big thanks on this. What we have done was that we have performed the first in human trial in patients scheduled for total knee replacement in a randomized placebo-controlled multiple and single injection dose. The reason for this was very simple. We are talking about a growth factor. And if the patient is already scheduled for total knee replacement, if something weird in terms of safety would have happened, it has not happened, would have happened, the joint nevertheless was scheduled to be taken out on this. So we have looked in this in multiple injection and single injection like that, and the follow-up was up to week 24. There were no differences in adverse events after intrathecal injection with Splifamine or placebo. We have no detectable systemic exposure. Most important for me, from a translational point of view, we have confirmed the mode of action in these late stage OA patients. In that histology and immune histology stimulated, we saw stimulated chondrocyte proliferation after Splifamine treatment. And we saw an improved biomechanical cartilage properties by using some ex vivo settings in this. In the samples we got from the study, like that, these patients were scheduled for total knee replacement, and so we got the samples and can investigate them. To our surprise, we saw a reduction in total knee replacement surgeries after therapy with Splifamine in the higher doses, especially the 100 microgram dose in this. So we don't have all the samples from this, but we have looked in the samples. Then we have run phase 1b, the so-called proof of concept trial. In a slightly different patient population. The patients were less severe osteoarthritis. It was mostly KL2 grade. And we have again looked for single and multiple injections. But in this case, we have used 2 cycles of treatment. So Splifamine is given once a week for 3 weeks. That's 1 cycle. And then it's repeated. In this. So we have looked in this so-called proof of concept trial in 2 cycles and followed up the patients up to 1 year. What we have seen is, sadly enough, we missed the primary endpoint. The primary endpoint was the medial cartilage thickness. But we saw dose-dependent decrease in loss of the total cartilage thickness, and we saw a significant effect in the secondary endpoint in the lateral compartment. So the medial compartment, and Henri was alluding to with the Galapagos compound, that is could be one of the major focus in drug development. The central medial cartilage thickness was not significant. There was a signal, but not significant. But we saw significant changes in the lateral compartment. And most importantly, since we are talking about a phase 1 or phase 1b study, we have not seen significant differences in adverse events. So the compound seems to be pretty safe in the whole setting. That was the point where the company decided, okay, let's go for a phase 2 trial, a bigger trial with 2 dose levels. The patient population was KL2 free in this, so a little bit more shifted towards more severe osteoarthritis. We have chosen at the time 2 doses and looked for the dosing regimen every 6 or 12 months for the cycle. We have treated the patients for 18 months and the primary endpoint was at 2 years, but the patients were followed after this 18 months in therapy, were followed up to 5 years. What we have seen is that we have seen significant dose-related increase in total cartilage thickness. We have seen a reduction of total knee replacement surgeries in the high dose group, means the 100 microgram group. But we have not seen an improvement in symptomatic readouts. Disappointing. The phase 2 was, I must add here, was not designed to look for the WOMAC or WOMAC pain as a primary endpoint. The primary endpoint was the total cartilage thickness, so a structural endpoint that was based on the very, very old FDA guidance and some recommendations from EMA or national advisors that at the time the study started, there would have been a possibility to get an approval based on structural improvement only. In the meantime, I think everybody here on the call is aware in 2018, the FDA changed the guidelines. EMA was not changing the guideline, but they are following this. They both agencies are now requiring structural plus symptomatic changes to get a disease-modifying claim in the label. That was happening during the runtime of the study, so okay. We looked then afterwards, in light of this change of guideline, we looked in the data in more detail, and I think that's what we heard so far in all the talks. Everybody is looking maybe in post-hoc, maybe in predefined analysis, in some subgroups, if you can identify a signal in one way or the other. And we have identified a so-called subgroup at risk. What does it mean at risk? At risk means at risk of fast progression. Fast progression not defined within 12 weeks, 24 weeks, but fast progression over the time with 1 year or 2 years framework. So in this subgroup at risk, defined by a certain joint space width between 1.5 and 3.5 millimeters, radiographic measured, and a significant pain level above 40 out of WOMAC pain 40 and above on the 100 scale in this. And in this subgroup at risk, so rather advanced or very advanced patients, we saw, first of all, the structural benefits still happening. So we see a dose-dependent increase in total cartilage thickness, but we also saw a clinically meaningful benefit in terms of WOMAC pain. The increase at 2 years was roughly 10 to 12 points on the WOMAC pain on a 100 scale. In this study, we have also looked for some additional points. We looked for PD biomarkers. to confirm the mode of action, and we were able to demonstrate this. And most important, what I think, apart from demonstrating a therapeutic efficacy, we have demonstrated in this rather big trial, 549 patients, you see it on the slide, we have not seen a significant difference in adverse events. So the compound looks pretty safe. And that's something one needs to, in my view, to be considered in the drug development in osteoarthritis. We are talking about a chronic disease, and we are talking about not only a very short treatment period, 12 weeks, 24 weeks. We are talking about, if we talk about a disease-modifying drug, of a long treatment in an elderly patient population. And so the adverse events for me is always something, you see it on the slides continuously repeated. That's for me one of the important things in a trial. Does not help if we have a good therapeutic efficacy, but we have a lot of adverse events, and we just this morning saw some press releases also in this. Lee can further allude to this and in this. So in a nutshell, what the data suggest with Splifamine, we have, first of all, and that's on the top of the slide, we have a favorable safety profile. And I have put the FDA OA draft guidance in the middle of this. It does not exclude the EMA or NMDA or whatever agency in this. But in the FDA guidance, there is clearly the statement in the ultimate goal of treatments is to avoid or significantly delay the need for joint replacement. So we need to deal with the complications of joint failure, reduce the deterioration of function and worsening of pain. And that's also what we hear in patient advisory boards. Arthritis Foundation has made this symposium together with FDA, and there's this white paper. That's also a very important point to consider, the deterioration of function or the cartilage. Symptomatically, we can address, as of today, quite a bit of the osteoarthritis pain. I say quite a bit, it's not completely controlled. But the most worry is the deterioration of function or the deterioration of the joint going to total joint replacement. I will be killed by the orthopedic surgeons here on the call since they're making money out of this. But for the patients, is quite a burden to go to the total knee replacement. So here on the slide, put together what we have seen so far. We have seen in the first in human trial that we have a reduction of total knee replacement surgeries. We observed the mode of action in histology, chondrocyte proliferation in this material. We have also seen some early signals in MRI. And we have seen that the produced cartilage, produced in remarks, accreted cartilage has improved biomechanical properties compared to the placebo. In the two subsequent trials, in the forward, so in the phase 2 and in the POC trial, we confirmed that we have cartilage accretion. Quite important in my viewpoint, without joint offloading. A lot of surgeons are aware, and there are studies out, also mainly in the Netherlands. If you offload the joint, then you get also some cartilage accretion. In the forward and in the POC trial, so in the phase 2 and the phase 1b, there was no offloading. The patients were walking completely normal and have a normal life in this. That's quite important if we talk about the medial compartment, since like my good friend Hans, the medical lead, used to say, the patients are walking on the heads of the chondrocytes, of the newly generated chondrocytes, and therefore most probably in the medial compartment in the POC trial, the primary endpoint was not met. In the forward trial, we saw cartilage accretion all over the joint. And I think that's, for me, since we are talking about a little bit also of lessons learned, we need to look for the whole joint, despite the fact that we have a cartilage, a bone, a senescence addressing agents. What have you. You need to have a look in the whole joint, since that's the organ that is actually diseased. Moreover, we have confirmed the mode of action using biomarkers, straight to my heart as a translational person. So really coming from the preclinical data, where we have used severe osteoarthritis. Let's not forget, if we talk about cartilage explants in the preclinical settings, this material is coming from patients with total knee replacement, so quite advanced patients. So we saw in the preclinical data the strong anabolic effect and the proliferation of the chondrocyte, and can now demonstrate this into a clinical setting. So really talking about translational science in this. And that's the point where we stand. Phase 3 should come up in this. Phase 2 is finished up to 5 years. And that's the Splifamine story.
Well, I want to thank you. Quite a story, and what a journey. Okay, so we'll hold questions and go ahead now with, I hope that our last panel speaker, Matthias Schieker, is with us now. And he is a senior executive at Novartis, who also has been involved in inflammatory diseases. And what I have asked Matthias to talk to us today about is his journey with the interleukin-1 receptor antagonist. And so, Matthias, the floor is yours. Thank you. Thank you.
Thank you, Nancy, and thank you for inviting me. It's a great honor. And as you said, Nancy, I'm here to give a bit of an introduction about the paper that we published in August on this contest analysis of IL-1. But before I do that, I quickly want to give an overview. And Chris outlined like the key parts of key important pathways to address, or in a way eloquently and nicely. So there's not too much to add. But at Novartis, we actually have a history in OA, and the ones of you who have been in the field for quite some time know that 15 years ago, or even 20 years ago, Novartis worked on COX-2 inhibitors, on cathepsin K inhibitors, or Simon calcitonin inhibitors. And so we had a large group actually within OA. And now, since about 5 years, we are the new kids on the block. I've been with Novartis now for 5 years. We are restarting our efforts within OA. So we are very early on in the pipeline. I'm overseeing an early OA pipeline where we try to, as Chris said, there's various aspects. There's the anti-catabolic approach, there's the anabolic approach, and the anti-inflammatory approach. We have compounds in the anabolic. You might have read the press release that that Merck out-licensed the anti-ADAMTS5 nanobody to us. And we obviously are interested in understanding the anti-inflammatory approaches with anti-IL-1. And the reason for that is about 10 years ago, so in 2010, 2011, we did at Novartis a trial with canakinumab, an antibody injected into the knee of OA patients. And this was more or less in the same time frame with the anakinra study, the Amgen study, AMG 108, the lutikizumab anti-IL-1 alpha beta antibody. So there are 4 approaches to address IL-1 in clinical studies. And honestly, all of them failed to show a pain effect and a function effect. And structures, anyway, very difficult to show for IL-1. So I think the field really from preclinical work, we all know that IL-1 plays a role in OA, and there's a clear evidence preclinically. And I think the big disappointment is that we haven't managed to show an anti-IL-1 effect for patients to benefit on pain, function, and structure. And this is what is out there. And we had the opportunity that Novartis invested into a cardiovascular trial, CANTOS, cardiovascular outcome study with 10,000 patients. And it's a study follow-up for 5 years, treated with 3 doses of canakinumab, the anti-IL-1 antibody, 50, 150, and 300 milligrams subcutaneously, and in, I said, 10,000 patients with cardiovascular disease. So these patients had to have heart attacks, had to have elevated hsCRP as a sign of systemic inflammation. Most of them were pre-diabetic. So these were patients with an increased risk or an elevated risk of inflammation. And so we had the opportunity to dig into this data set. So we did a secondary analysis in this data set to actually look at OA. And what we did is our primary endpoint was the time to first incident neo-hip replacement. And the second, so really outcome joint replacement analysis. And the secondary endpoint was time to first occurrence of an AE-related adverse event. So in a sense, AE showing symptoms of osteoarthritis. And this analysis clearly showed a delay in the need for joint replacement. So when you look at the paper, the curves separate early on, even after a year. So we had here really the opportunity in a huge patient population with a long treatment duration to look at the joint replacement and to show an effect. And all treatment arms showed an effect, a reduction of joint replacement and a reduction in the adverse event outcome. So we know the limits or the limitations of the study. First of all, it's a secondary analysis. It was not pre-designed to look at OA patients. We don't have any imaging data, so we don't have structural data. We really have the joint replacement and the symptom analysis. And 40 to 47% actually reduction of joint replacement. That's quite a number. And so there is a clear effect shown in this analysis that IL-1 reduces joint replacement. And so this is actually what I wanted to give here as an introduction. Happy to answer any question, but I think, Nancy, the intention to bring me to this panel really I want to, hopefully was that I want to add on to, as said, the anti-catabolic, the Wnt inhibitor, the anti-senescence, the anabolic approaches, somehow the bringing IL-1 back, anti-IL-1 back to the table after 4 failed clinical studies, and yeah, open up for discussion. Happy to answer any questions.
Yes, Matthias, thank you very much. And you're right. We wanted to show everyone where the different modes of action were of the compounds and the studies that had been done to fill the gaps. That's exactly right. Okay, well, thank you very much, panelists. And now I'm going to turn this over to Dr. Lee Simon, who's going to ask you all to join a panel and take some questions from the audience. And a few questions have come in, but I'm going to leave those to Lee to bring up. Okay. Thank you, Lee.
Well, thank you very much, Nancy, and thank all the speakers. And first, I'd like to ask if anybody online has any specific question to ask before we, in fact, I can ask some things, but let's see if there's anybody online who is interested in asking our speakers something. And Jason, how do they do that?
Yeah, Lee, if anybody wants to ask a question, they can just turn on their mic and camera. I think Kale was one of our first people that asked a question. So if Kale, you'd like to ask it yourself, we'd be happy to have that.
Yeah, thank you, Jason. The question I had was really for the group. First, thanks for the
fantastic presentations. I think it's been repeatedly shown that OA pain is more related to psychological conditions like depression or anxiety or pain coping strategies than to radiographic severity. However, it doesn't seem that industry trials to date that are looking at different OA treatments have not really included psychological or psychosocial measures as covariates in their analyses. Could such an approach perhaps give a clearer indication of whether these treatment options are able to lessen OA pain and improve patient-reported outcome scores?
Could you comment what kind of specific outcome measure would you use for this quote-unquote psychological assessment? What are you referring to exactly?
Well, I think there's a number of different things you could look at, but just I know that depression and anxiety specifically, so if there's questionnaires about that, or any number of mental health questionnaires, pain catastrophizing, self-efficacy, there's a lot of different things that could be looked at, but it doesn't seem that anything has really been taken into account. And I wonder if that creates some of the variability that we see with patients, and then that makes it in turn more difficult for these different treatment options to achieve a therapeutic benefit over placebo.
You're right. It's a very difficult problem. And some of the outcome measures have been secondary outcomes of looking at depression and catastrophizing. However, they have not been elevated to the point of being an important approval outcome measure, when most of the work that we've been looking at has been for drug approval. Chris, do you have a comment to make about this?
Yes, Kale, great question. And Lee was already alluding to this a little bit. So what we have used some questionnaires like PGA or SF36 in this context, and I'm pretty sure my colleagues from the other companies have also looked in this. One of the points is, Kale, really what, at least in my experience, and everybody can comment or criticize in this. OA patients have this kind of psychosomatic stage, maybe catastrophizing and the whole thing. But what we see, and Ben was also alluding to, what we see in the trials, the patients, and that's also coming out of patient advisory boards, what we heard, the patients are desperate to get a treatment in a certain way. So this needs to be taken into account if we talk about this psychological readouts. They want to come in the study. There's a very nice statement that I still like, despite the fact that it's some years old, from Elena Lucina from Boston. The time is a healer. So the patients are coming in, and you can see this very clearly also in observational studies like the OAI. The patients come in, they have a high pain level, they have anxiety, they have depression. They come in the trial, even non-interventional, and they feel better shortly after. And that's a confounding factor. But sure enough, Kale, it's a very good suggestion to further include and really look at the status at baseline of the patients about their pain reporting, about their anxiety, depression, all these kind of things. I think that's worth to have a very, very close look in this, despite the different OA phenotypes in terms of disease pathophysiology.
This is Matthias.
To other people, go ahead.
So
Matthias?
I just wanted to highlight that this is a very, very important question. And so see, we are doing our early studies with less than 100 patients. And if you want to measure pain effect, you need to be clear what you're measuring. So that's more than VAS pain or WOMAC pain. And you need to understand this very clearly. So there are various factors that influence pain measurements, as you highlighted and as you put in chat and said. So this is important because if you want to detect an effect on pain, you need to be clear what we're measuring. And this is not only for OA, this is for other diseases as well. We look at tendon disease. So all musculoskeletal disease need to be very, very specific. And there are activities ongoing to address this. And I only want to say I fully agree. If we want to measure pain effects, we need to know what we're measuring, and we need to understand how pain measurements are impacted by various, and as you mentioned, depression or other diseases. And so I only support what you're asking very much.
And Kale, this is Nancy Lane. I wanted to harken back, and this is going to reveal my age a little bit. But when we did the very large chondroitin sulfate study, the GATE study, one of the exclusion criteria was depression. And they thought of excluding patients with depression or taking any mood altering type of medications would try to reduce that confounding effect. And we all know the results of the GATE study. So let's just say we haven't quite figured out exactly how to do it yet.
And Elena Lucina, you have a comment to make?
I think this is a great discussion, and I think you're raising $100 million questions about how to address the contextual effect and direct treatment effect and total effects. And I think, what we all study in trials, because we're scientists, is we're focusing on direct treatment effects because we would like to know whether the treatment actually works better than placebo, what are direct effects. But from clinical point of view and from patients' point of view, what they really care is about total effect. And they really care less whether parts of it is contextual or placebo effect or some other components. And so I think that's where the science and the practice sometimes do not come together. So I think as scientists, we should continue to do rigorous trials and try to look at the direct treatment effect. But I think we do need to realize that what patients are looking for to get better. And that's the challenge. So I think studying placebo effect and understanding its component and recognizing that that is a part of what patients kind of will receive as a package, no matter what treatment they're getting. If when we're talking about subjective or patient-reported outcomes, that's an important part.
The problem of expectation bias is an enormous problem associated with the placebo effect. And there are other aspects of that that are also very important, as Nancy has mentioned. Many years ago, we did a bisphosphonate trial looking at the changes in subchondral bone. And we had to select 23,000 patients to find the 2,300 that we would study. The heterogeneity of this disease is enormous. So for us to understand changes in structure, we have to think about what Novartis did at a 10,000 person trial over multiple years. We have to think about, or selecting patients that'll be more progressive. Does anybody have any idea about how to go about doing that at the get-go of the trial to select the patient that within the window of opportunity will demonstrate change that's meaningful? Any ideas?
Very challenging question, Lee, if I might chime in this whole thing. I think one important point, and Elena, sorry I missed you, I've not seen you in the list of participants, but what Elena mentioned very clearly, this contextual thing in the clinical trials is confounding, at least in randomized clinical trials, quite a bit of this. What maybe I'm provocative, but what I always think, and Lee, it's a little bit to the point you just asking, what I always see in this data, and Ben, Jeymi, have shown the data even in this context, the patients become better overall, even on placebo. If it's contextual, and Jeymi has had a very nice poster or a presentation, whatever you want to call it, in this series, in the OACS series, it's more contextual. So the patients, even on placebo, become better if we talk about patient-reported outcomes. They are not necessarily become better in terms of structural outputs. But if we talk, and I'm not a doctor treating patients, but if we talk about daily practice, then patients become better with an injection, even, and Jeymi showed this on his poster talk very nicely, even with a sham operation. Isn't it worth to loudly think about if this is not worth something we should also have a very close look on?
Yes, but this is Nancy Lane, and I want to say that this has always been is a big challenge in OA trials. But on the other hand, I want to say something that's pretty important. OA pain usually comes and goes anyway. It is a disease that has flares and times when the disease isn't as active. And we all know that. So we also have to factor that in. Just some pain, you flare, it goes away, or it reduces. It'll come back, but it reduces. And we also have to think about that as we find better instruments to measure the outcomes.
Yeah, good point, Nancy. It brings me to the link, and Lee already touched a little bit upon this. The link between the readouts that we have as hard facts, like MRI, X-ray, what have you, and the patient-reported outcomes. Let's not forget a long time, and I'm for quite some time in the field, a long time it was mentioned bone marrow lesions. Great. We are on the way to demonstrate with an imaging technology with the bone marrow lesions, the pain readout. No, they are coming and going. They are fluctuating like the pain reported outcomes. So that's something. But how to build this in, Nancy, to be honest, how to build this in a clinical trial protocol in a way that you get a meaningful readout afterwards. That's for me the question.
That's where we need to come together. I totally agree with you. We need better readouts.
Yeah, I think the issue is, and I think, and Chris just mentioned this, that when you invite people to participate in trials, they're more likely to come participate in trials when they do have an active flare. And so when the flare passes, then they get better, and they get better probably with treatment and without treatment, because as Nancy said, it is a disease of flares. So the trick is trying to distinguish between flare and background level of severity of the disease, and try to figure out how to not to measure baseline at the time of active flare, because once we do this, then we may see very controversial and unexplainable results. So maybe some sort of a washout period, or everybody gets placebo for a certain period of time when the flare is going to level off, and then starting one arm on active treatment and another continue on placebo potentially can minimize that effect. But I think that that's what's interesting and challenging about doing trials in a way, because it's
and the other piece about that, Elena, is for us to think a little bit about non-traditional instruments. And it could be that monitoring steps per day, or the wonderful activity monitors could end up telling us a lot that will be important in terms of determining the efficacy of an agent. So we also have to think creatively there.
So we have to think creatively about the trial designs. So Elena raised the issue about a lead-in for placebo, but another alternative may be a randomized withdrawal trial, where everybody gets the drug up front, and then you randomly withdraw, predicated on trying to understand either flare as symptom basis or even structural change. These are much longer, complicated trials, but this is a much complicated disease, despite being mostly symptomatic. But if we're going to do structure, we have to think about that. And Jeymi, did you have a comment to make?
Yeah, I think I've got a couple of things I'd just like to add, I guess. First of all, I going back to the sort of original question, looking at the chat, I kind of disagree that pain and radiographic severity aren't related. I mean, we have that work from Tahina Yogi's, now it's what, 10, 12 years old, the study where she showed association between more frequent unilateral symptoms, subjects with more unilateral symptoms, frequent symptoms associated with increasing severity for both the OAI and the most cohorts. And we've shown with our data that subjects with unilateral symptoms can discriminate their pain more, especially, and this comes to the other part of the question, when you use a questionnaire like we use the WPI and symptom severity score, and that's a fibromyalgia diagnostic criteria questionnaire. And we spoke to several pain specialists, and I know Lee and Nancy are both very familiar with this, with the work we did. And we identified a level where we can take out, or we can screen out patients with potentially pain confounding perceptions. We also took out patients with depressive disorders on major depressive medication. And we found that isolated, you can't perfectly isolate the pain signal, but we managed to show discrimination on pain. And I note that when with the Splifamine data with Christoph's at-risk population, they did similar things. The other part that we did was we limited the joint space width to between within a fixed limit. In our pivotal studies, is 1.5 to 4 millimeters, and this showed that greater responsiveness with our radiographic measures, and also a connection with our WOMAC pain and pain NRS signals. So I think that, you know, we're getting there slowly. The field is moving forward. We're understanding more about the nature of OA pain and the placebo effect. And so with more time, we're going to get more of a homogeneous trial populations across different manufacturers, which will hopefully isolate pain signals more. So I think, you know, on the whole, they're kind of some of the from an industry perspective, we have to keep commander's intent. We have to figure out how we are to get that approval. And so we have a slightly different outlook on what our goals are from just purely the academic research side of OA as well. Obviously, we're very interested in that as well, and we continue to help hopefully push back some of those boundaries as well. But all of our trials are connected to getting that goal, which is the approval. The only other comment I want to make regarding very quickly about the placebo response is that because of the contextual effects and because they're strong, a treatment has to show a benefit above that to be a treatment, you know, so that's the only other comment I'd want to make.
Tahina, you have been mentioned. Martin, you'll be next. Tahina?
Yeah, I don't have that much more to add other than, you know, I think OA is complex, as we're all stating, not just from a pathophysiology perspective for structural disease, but also because of the multifactorial nature of pain. And I completely agree with Alina's comment that patients are looking for that total effect, what's going to make them feel better. But from a business practical perspective of approval, you have to show that it's better than the comparator arm. And, you know, I think this ties in very well with the next session on OA phenotypes, though we're not going to speak specifically about pain phenotypes. But I think, you know, the strategy of who to enroll and some of these strategies that are being used, in addition to really seeing if our symptom instruments are fit for purpose. And I know we've talked about this at the ACR OA study groups over the past 2 years, over the past 2 conferences, about whether or not WOMAC pain is really the best instrument to look at changes in pain over 2, 3, and 5 years. We know that patients accommodate to their symptom level, they alter their activities, and what's a 4 out of 10 pain today is not the same necessarily as what 4 out of 10 means to them 5 years later. And, you know, so I think we have a number of challenges in measuring pain, self-reported pain, and understanding if we're doing it properly? And also, are we able to capture that portion of pain from osteoarthritis that is separate from all of the other contributors to pain?
Martin.
Yeah, I would thank you for your presentations and also for the discussion so far, which is really great because it identified challenges in the tools that we're actually using to measure pain, patient-reported outcomes, as well as actually structural outcomes. In more of a general picture, I would like to address the question in a clinical trial, how to define the primary outcome. How do you choose whether you will have a structural outcome as your primary endpoint? How do you versus a PRO-related outcome? And this has actually been a theme in the presentations and also in the history of some of the trials that were presented, like, for example, the UBX 101 trial was using a pain outcome study, although the drug by itself originally was developed, to my understanding, as a disease-modifying drug. And in the case of Samumed's drug, perhaps Jeymi and Ben could comment on this, it was tested in some studies with structure as a primary, and in the currently ongoing study with pain as primary. And Jeymi, perhaps you could also comment on how you would approach this in your NDA. So the general question is how to navigate this in the current regulatory environment. And the question following on that, and an even more broader question, is does this even make sense for us if we're trying to develop a DMOAD, that it really has to have this direct effect on patient-reported outcomes?
Yeah, so that's the $6 billion question, probably.
Someone had asked it. So.
The part of this is historical because before the 2018 revised guidance from the FDA. First, before any of this, really, as manufacturers, we are beholden to the FDA or to any regulatory agency as to what they require. So it's kind of set to us. And before the 2018 draft guidance, the old guidance in 1999 only stated a change in medial joint space width as being enough to be to gain a label as a disease-modifying drug. I can't remember the exact terminology, and I'm sure Lee would know, but it was essentially that. The 2018 guidance made it clear that there had to be an effect, a patient benefit, an effect on how a patient feels, functions, or survives being joint replacement, and they had to be tied in together. We've had these discussions with the agency, and they've made that very clear. And as a result, that means that pain, without a pain endpoint as your primary, you can't get an approval. So we had to set that as a primary. Then after that, the next problem is defining what is the imaging that will reflect that structural change and also tie into a patient-reported outcome benefit. And that's proving difficult, and I think all of the panelists will agree with that. We've continued to look at X-ray because so far MRI has not been shown to be any more than a biomarker, which X-ray has been as well. And there are initiatives like the FNIH, which hopefully will, they're entering imaging modalities and measures into the biomarker qualification project, which is run by the FDA, and they will hopefully get us more clarity. But time is the enemy of the manufacturer, and so with time, hopefully we'll see these, but it means that from a regulatory strategy, certainly from the Samumed perspective, we've decided to go for pain as a primary endpoint because we know that's an endpoint that if we meet, we will get an indication. We are submitting a lot of other evidence for structure as well, so we won't take it off the table as a potential component of our label, but we don't know what the FDA will, how they will consider that. And the final part is function. People have mentioned this, and it's kind of like the forgotten stepchild of the endpoints. And you know, the FDA have said that potentially pain and function together might be enough to gain a treatment of OA indication. So, so how we measure function and how we demonstrate that to the FDA is something that we should, maybe as manufacturers, focus more effort on.
Henri, do you have a comment to make?
Yes, I hear very interesting comments, and I read very interesting comments as well. I think as drug developers in the field of, let's say, structure modifying drugs, and I take our own mode of action on cartilage, it is really very challenging to show at the same time, in the same time lapse, a structural effect and a clinical benefit. It is not unlikely that clinical benefit may follow structural benefit, but it takes time. And as Jeymi said, time is in fact the greatest enemy of developers. I hear also opinions from experts saying, as long as the agencies look at OA like they look at RA, we face that problem, that they expect that we can show altogether structural benefit and clinical benefit. Whereas, for instance, in the field of osteoporosis, they agreed with a surrogate endpoint like bone density. So that's one big challenge for us. Another big challenge is indeed the assessment of the clinical outcomes and the relationship between the patients and the tools that we use and whether they are appropriate. And I think that I'm dreaming of let's say, a joint, let's say, approach with all industries involved and with the community involved to look for more new technologies to measure pain and clinical benefit in a more real life situation. And the technologies are there. The big challenges are the validation of these technologies and convincing the agencies to look at these technologies as being valuable. So I think there's quite some work to do and quite some dialogue needed with agencies to find common grounds of understanding how these drugs, these new drugs that try to modify the structure and to try to have an impact on the natural progression of the disease, how these drugs can be, let's say, marketed, maybe with not immediate clinical effects, based on quite objective measures like X-ray or qMRI and others. Yeah, I think there's a great work for us ahead and a great deal of discussions with agencies in that field.
It's also important to recognize, and Dave Felsen has got a note in the chat box that actually points out that the variability of pain response. And just so that everybody knows, there is a public-private partnership going on between the FDA and a group called ACTION, ACTTION, to improve pain outcome measurements, to figure out how to actually select patients for clinical trials that can consistently give repeated pain responses that are not variable over the time of a clinical trial. David, might you have something to say about this?
Yeah, Lee, just to second what you just said, but I agree. I put it in the chat. I mean, I think the rate of placebo response historically has been increasing in OA trials, and that's made it really challenging. This is both in RA and in OA. Really challenging to see effects of treatments that reduce pain, because the treatment effect statistically is, compared between active treatment and placebo based on the standard deviation of that effect. And the wide variability of a placebo response in terms of pain generates, very wide standard deviations, makes it very hard to evaluate this. So there are strategies, as Lee just said, to train the patient, for example, to do run-ins or pre-randomization tests to find out whose pain is tremendously variable, to exclude them or train them into how to, sometimes it's because patients evidently just aren't that observant of how much pain they're having with particular activities. And if you sort of train them to be more observant, they're more consistent. And that makes it a little easier to detect the effects of treatments on pain. But I can't see regulatory agencies or the clinical community being very enthused about treatments for OA that don't have any measurable effect on patient outcomes. I mean, that seems pretty unlikely to me. That same argument, by the way, is going on in rheumatoid arthritis with remission, and I think many people are reacting against definitions of remission, which seem to want to exclude patient-reported outcomes. It just seems pretty unlikely. I mean, we're there to treat patients and make them feel better, and our challenge is to figure out how we measure how they're feeling so that we can detect effects of treatments that do make patients feel better.
Yeah, but David, if I may directly chime in here, very good points, and we talked about this several instances, and I think the recommendations or some of the ideas of Nat during this OACS session that you were mentioning were pretty good to look for this. The point is really to convince then afterwards the agency that such inclusion or exclusion criteria can become forward for the approval of a drug in this. We have this fluctuating pain reporting and pain in the patients. It's challenging. We need to have a very close look in the clinical trials, and that's also coming to the point Tuina mentioned, maybe in the next sessions with the OA phenotypes. What we learned over time, and Jeymi, Ben, Matthias, please chime in if you have further, Henri, if you have further comments. We are coming to the point to select the patient population very carefully in order to demonstrate an effect. And the selection could also mean that we are selecting for correct pain reporting, less depression, and what have you. That's a little bit of a lessons learned. But if I look for the journey that we all have had in drug development in osteoarthritis, if I look for the journey, we are coming from X-ray with a huge variability measuring the joint space width. We are now approaching more and more, and I'm still not 100% convinced that the agency will accept everything. We are approaching MRI as a more precise measure in my viewpoint. But that's a tremendously long journey. And if and to be honest, I'm a patient, I have osteoarthritis in both knees. If I'm, as a patient, looking for this journey, then most probably, I will be far beyond my retirement before anything is coming through on the market. Sorry, Lee, not joking. I'm a little bit younger than you, but not too much, I must say.
There are two things that have been commented on so far that's important to comment on. So many people measure function through the WOMAC 17 questions, and no one has ever tried to do a WOMAC function as a primary outcome that I'm aware of in an RCT therapeutic trial. And it is more sensitive than some of the other things like a stand up and go and a 30-minute, 30-second stand up and 50-foot walk time and things like that. And it would be interesting to see more data where function is measured or differently measured. The FDA has created an effort to try to understand using functional measurements like the watches and the Fitbits and things like that, and whether that will actually pay out over time, I don't know. There was another question about, could a withdrawal design, randomized withdrawal design, address the ethics issue? Well, it all has to do with rescue therapy. And if you were rescued, you failed the therapy. And so the ethics doesn't really play a role here when done correctly. But I do think that Jason might have something to say about our time. Jason?
You probably still have 3 or 4 minutes before I cut you off.
Good. Aileen Lettingham asked a question about measured physical function. Aileen, do you have a comment to make? Maybe not. No, she says no. Okay. But I think, Martin, do you have something further to make?
Yeah, I just wanted to make a quick comment about the regulatory environment. So we obviously have seen changes made by the FDA in their guidance, and our current guidance is that there has to be a patient-reported outcome benefit even in a trial of a drug that was primarily developed as a DMOAD. So as a field, and especially actually today here with this opportunity having scientists from industry and academia, the question is how strongly are we enthusiastic about these current guidelines, and is it really a valid concept to expect symptom modifying benefit as well as DMOAD activity? I mean, we have reasonable, of course not perfect, pain medications, and wouldn't it be at least worth a thought to approach the regulatory agencies to again separate these outcomes and maybe even include an approved symptom modifying drug in combination with a DMOADs to be developed.
Well, maybe actually Jeymi can give us their thoughts about this, since they're trying to do both at the same time. That's really a big deal. So Jeymi, what do you think about that?
Yeah, so I think that we carefully went over the guidelines, and we've spoken with many people externally as well, and in fact a lot with you, Lee. And you know, really, I mean, ultimately, we agree with the FDA's position. And there are 2 indications now available from FDA. One is treatment of pain of OA, and the other is treatment of OA. Treatment of OA implies treatment of more than pain, and you can put that, hopefully, if you've got enough evidence that FDA consider sufficient, then different parts of your label can reflect that data. And there is still actually a great unmet need for pain medications. I mean, there's only really non-steroidals, corticosteroids, and now we have, you know, the anti-NGFs potentially, but they're serious drugs with some serious adverse events. So there's still a space, and we've seen what's happened with the opioids as well. So there's still a great space and great unmet need for new pain medications that are safe. Our strategy has been, let's go for what we can and put in everything we can to try and get that treatment of OA, but ensure that we safeguard pain as much as possible. And then with time, you can always build your label. So we know that osteoarthritis takes a long time and to progress, and even for rapid progressors. And so we know that with time, we can hopefully, if we don't have enough structured data now, we can build that, and we will see if we will meet whatever the FDA require. The other flip side, of course, is if you don't have an initial pain signal, then you have to build your evidence to show that your structure change then leads to a symptom change, and that's a harder boat to be in. Potentially, there's the accelerated approval pathway, which may allow you to get a conditional license or label and then show that you fulfill those further endpoints. but we've not had those discussions with FDA, and maybe some of my industry colleagues have had those discussions and can elaborate more on that.
Thank you, Jeymi. I've been told that we're at our time point, and I want to thank everybody, the organizers asking me to participate, as well as Nancy and all of the speakers that we've had, and everybody else that's actually joined in asking questions. I think this has been a very interesting discussion. And in the end, we really want to do better for our patients. And I think these kinds of discussions lead us to a better route for that. So thank you all very, very much. And
Yes,
Jason,
thank you. And now we'll give it back over to Jason. Thank you all. Thank you, Lee. Thank you, panelists. Thank you, community.